Hello, I'm going to be giving a report on the activities of the DevoWorm group for 2022. The DevoWorm group is associated with both the OpenWorm Foundation and with the Orthogonal Lab, and so that's why I'm presenting it at our open house here. The OpenWorm Foundation annual meeting is in mid-November, so I'm going to be presenting this to them as well as an update. You can see our website down there at devoworm.weebly.com, and we have a GitHub repository as well. So our first activity that I want to highlight from this year was the Google Summer of Code, and it was very successful. We had 2 projects with 2 students each. We had the Digital Microspheres project, and that project sponsored Karan Lohan and Hari Krishna Pillai. They're both from the same university in India. And the Digital Microspheres project is concerned with building a spherical computational model for embryo data. So it's taking 2-dimensional embryo data and tiling it onto a sphere. And we'll see an example of that in a little bit. The second project was developmental graph neural networks. And this is a pipeline for the DevoLearn platform. So the DevoLearn platform was something that we developed in years past, and it's been released as an open source project, an open source platform. And this project concerned building graph neural network embeddings that interface with that DevoLearn platform. So the DevoLearn platform would allow you to segment image data or microscopy data, and then the graph neural network would involve representing it in some way. And so we had 2 students here, Jiahong Li and Wataru Kawakami. And these 2 students, one was from, I think, Hong Kong, and the other one was from Japan. So congratulations to all of our Summer of Code students. So the Developmental Graph Neural Networks project, the shorthand being D-GNNs, this was something that we developed over the course of the summer. We had a pipeline that we developed from segmentation, which took a lot of elements from the DevoLearn platform and refined them in a lot of ways. So there were a lot of pull requests to the DevoLearn repository that involved upgrading these capabilities. We had not only our Google Summer of Code students, but other people as well involved in this process. We also had a second phase, which was replicating graph neural network embedding found in a paper that one of the, I think Jiahong identified, and then reconstructing, refactoring that model and using it in ours. So this was all an open source effort from open source code, and we were refactoring that code, plus refactoring the DevoLearn code. And so you can see down at the bottom right, there's this TT-GNN, which was used for the GNN embedding. The overall structure of it is right here in front of us. There was a challenge of finding mapping information from frame to frame and building networks where you could predict the links from frame to frame as they change, because you're dealing with embryo data, which is constantly expanding. And so we also had, we presented in the weekly group, and I think they did a very good job. And so this is now available in the DevoLearn organization under the DevoGraph repository. So it's an add-on to DevoLearn, and you can download all of it for yourself. The second project was Digital Microspheres. So this was creating an atlas and analysis tool of the embryo's surface. So we're mainly concerned in this project, or at least for this summer, with the axolotl embryo. The axolotl embryo you can see on the left is a very large embryo, and it's transparent with respect to brain development. So it's a very useful animal model for looking at those sorts of transitions and development. One of our collaborators, Susan Crawford Young, built this ball microscope, which is a microscope where you put the embryo on a cover slip and you put it in the center of that sphere, and then all of those cameras on the edge, there are 9 points of view, you then start taking images of that stage, and what you get is images from all different angles of the same embryo. So you can see at the right, these are images from different perspectives. What we did in Digital Microspheres was then take data like that and map it to a computational model. So we took these flat surfaces, these 2-dimensional images from different perspectives, and then mapped them all to a sphere and registered them as you could see like the surface of the embryo. So now We have two rival models that we worked on over the summer. Both of those models involve taking the data, mapping it onto the sphere, and then allowing the user to explore that sphere in different ways. And so there was a lot of, this project involved a lot of sort of figuring out how to do this spherical mapping. We debated different algorithms, we debated different approaches, and I think the students did a very good job of getting that all squared away and building software that you can now download. We have a repository for digital microspheres in the DevoWorm organization. So we've also been involved in the virtual conference circuit. So switching gears to research, we've done a number of conferences this year. One of those being NetSci and Network Neuroscience. So that was in July of 2022. One of the main NetSci session, our talk was hypergraphs demonstrate anastomoses during divergent integration. And so you can see a picture at the right of what that means. We were modeling the embryo with hypergraphs and then showing over development that development is this process of going from an undifferentiated embryo to something that has structure, something that has organs and tissues. And those organs and tissues are joined together through anastomoses, or these little sort of bridges between the tissues. So in the heart, you have anastomoses between the chambers of the heart. In other organs, you have anastomoses that join things together through small pores or things like that. And so in networks, you can represent those ways of connecting the subgroups. So as the embryo differentiates, you get different subgroups that emerge, and those subgroups are also still connected through these anastomoses. And we put this in the language of network science. So this was something, we have it on our website. We have a link under the media heading. So if you want to check out the video of that presentation, you can do so. We also attended the INCF assembly in September. And this was a paper on agent-based modeling that was actually from the orthogonal lab. This was an introduction to developmental embodied neural simulation. The reason I put it under DevoWorm as well is because it did involve a lot of insights from development, and it involves agent-based modeling and embodied models of behaving organisms. So this is very relevant to OpenWorm and DevoWorm. And then we attended the SciAm workshop on network science, or rather I did, and this was in September of 2022. And this was a presentation, another version of the hypergraphs talk, but this was embodied hypergraphs, analyzing interactions in time-dependent systems. And so this was this was actually going a step further than the NetSci talk and talking about the embodied aspects of hypergraphs representing embryos. So what I mean by embodied is that you have a head and you have a tail and you have sensors on the edge of the network. And then you have a sensory system, you have organs on the inside of the network, and that network wires itself according to its embodied context. So it's a very interesting approach to this, application of network science and approach to the problems of development. We also had our weekly meetings. We continued on with those. We meet generally Mondays at 10 a.m. Eastern time. This is in North America. So if you're in other parts of the world, just figure out what Eastern time is relative to you. This is announced every week in the Slack, in the OpenWorm Slack, in the DevoWorm channel. And we usually have a pretty decent turnout. We have several people at least. And we do a lot of things in these meetings. They're about an hour and 20 minutes. We have topical reviews. So we've talked about a lot of different topics this year. We've talked about morphogenesis in its different forms, embryophysics, approaches to imaging, as we showed in some of the GSOC projects, systems biology topics, and developmental biology topics. The idea is to get people familiar with the literature, to get them, maybe some of the computationally oriented people, familiar with the biology and the like. And we always have our favorite topics that we run across papers, so these get presented in the topical reviews. We also have our biology and computation updates, which are usually small demos or presentations that people give during the meeting. These are open source projects, or they could be papers we're working on or presentations we're working on. We might have software demos. We might have things like collaboration management, where we review tools for collaboration, or review our GitHub repositories, some of our open issues. Finally, we have tutorials. So we talk about how to use Jupyter and Colab notebooks. We talk about model building and other technical topics, generally computational, but sometimes having to do with microscopy and data analysis or data acquisition. And so one of the innovative new themes we've been developing this year has been the cognition and psychophysics in cells and embryogenesis. So, we're looking at embryos, we're interested in embryos and how they behave and how they develop. And we're also interested in some of the other things that they do, like as they develop their behaviors as adults or as larvae in the case of the worm. And we've really attacked this from a number of different ways. So we actually have 2 papers this year that one is a book chapter, one is going into a special issue of a journal that kind of address this issue. So the first one is looking at the diatom Basilaria paradoxa. And so this diatom is a colonial diatom, means we have multiple cells in a chain, and it moves around in different ways. And so this movement is, we don't really know. We know the physics of the movements, but we don't necessarily know why they do this. And we know that they respond to stimuli, and we know that they have a behavioral output and they behave collectively. And so this paper was an attempt to talk about the psychophysics of some of their sensory world, some of the things that they do with respect to their environment and how they behave in a colony. And one of the concepts that we talk about in here is the idea of a collective pattern generator, which is like a central pattern generator, say in an insect or in a notochord or a vertebrate. But these are single cell organisms that live in colonies. So it's a very different process. But nevertheless, they maintain this rhythm and they maintain this collective behavior. And so this talks about this, among other many topics in the, from the psychophysics literature. And that's definitely something that we're developing. This is just like kind of our first foray into it. The second paper is this embodied cognitive morphogenesis as a route to intelligent systems. So coming from that talk to the SIAM conference or the SIAM workshop, we're interested in this embodied cognitive morphogenesis approach, which involves the structure of an embryo, so through something called tensegrity, or keeping dynamic tension within an embryo as it develops, and then how the embryo breaks its symmetry and exhibits fluctuating asymmetry. And these are important for developing different organs and different relations to the environment and different parts of the connectome. And then also how it interfaces with its environment as it's developing. So as it's transforming its shape, how is it interacting with its environment? And if you think about development as an embodied cognition, it actually opens up new avenues and sheds new light on the process. So it's a very interesting sort of new direction for us, and we'll be following up on that in the new year. So finally, I'd like to thank our contributors during this 2021-2022 period. We've got probably about 20 people listed here. Some of them have contributed to our GitHub repository through pull requests. Some of them have attended our meetings. Some of them have been Google Summer of Code Scholars. And I think all of them are on our website under contributors. So thank you for your attention.